This video will be the first video on the control of gene expression in eukaryotes. Early on, people thought that regulation of gene expression in eukaryotes was going to be fairly similar to what was found in prokaryotes. And in fact, there are many similarities. We'll find that there are transcriptional activator proteins, transcriptional repressors. We'll find that there are transcription factors that can bind to the DNA. And we'll find that those factors can respond to signals in the environment, such as effectors. A few years ago, scientists began to realize that there is a whole additional layer of regulation that is found in eukaryotes, and it has to do with regulation of the chromatin state. In other words, the accessibility of DNA for things to bind to it and for it to be transcribed. So first I need to introduce a term. This term is chromatin, and so chromatin is a word that refers to the DNA and all of the associated proteins that are found with the DNA. In eukaryotes, that consists primarily of nucleosomes. So nucleosomes are complexes of proteins, and around those proteins the DNA is wrapped. So a nucleosome is made up of 8 different proteins. The names of those proteins are histones, and there are 4 different types of histones. There are 2 each of H2A, H2B, H3, and H4. Often a nucleosome is depicted either as a ball, as shown here as this orange ball, or as a spool around which the DNA is wrapped. So DNA wraps around a nucleosome, and each nucleosome takes 146 bases of DNA, and the DNA wraps around that nucleosome 2 times. If you look at the size of a nucleosome wrapped with DNA, that would be around 11 nanometers wide. So now I'm going to switch to a different figure. So now the nucleosomes are shown as these little cylinders, these purple cylinders. So the DNA wraps around each nucleosome 2 times, and then there is the second level of organization of those nucleosomes. This second level is called a solenoid, and this is organized by another histone protein called H1. So H1 organizes 6 nucleosomes that are wrapped by DNA into what looks kind of like a spoked wagon wheel. This wagon wheel would then continue on, and it's called a solenoid because it's very much like a spring or a slinky. It will continue to wrap around, coiling around. When you actually coil the solenoid around and around, so each ring has 6 nucleosomes, it will create a new structure. This solenoid is 30 nanometers in diameter. After that solenoid level, we now have a third level of organization, which is the scaffold level. And so now the solenoid, that 30 nanometer diameter structure, is just depicted as this purple line. So the solenoid attaches at various regions to some proteins that are shown here in red. These attachment sites create little loops going out, and these little loops will then curl up among themselves, and this is forming the scaffold and the level 4, which is the supercoiled scaffold. The scaffold is 300 nanometers wide, and then the supercoiled scaffold is actually 700 nanometers wide. And this final organization is actually the structure that you see in a chromosome that is condensed during mitosis. So here is a summary of those different levels of chromatin organization, going all the way from naked DNA all the way to a condensed chromosome. I'd like to point out that it's really these 3 first layers of organization that are all found during interphase, and it's only during mitosis that that last level of organization is ever observed. Now, historically, when people have looked at cells under the microscope, they've done something called electron microscopy, and this type of microscopy looks at the density of different structures within a cell. And so this particular figure shows a nucleus. So here's the nucleus in the middle. And one thing that people noted was that there are regions that are more densely staining. These tend to be along the edges of the nucleus, and there are also regions that are more lightly staining within that nucleus. If you looked at where DNA was in that whole nucleus, you'd find that it is throughout, not only in these dense regions, but also in the lighter staining regions. Historically, scientists used certain terms to describe these different regions of density within the nucleus, and those two terms were euchromatin for the lighter regions and heterochromatin for the dark regions. It's now well understood that the regions that are euchromatic, or that are stained as euchromatin in a nucleus, have DNA that looks more like these regions here. The DNA is less tightly packed, and it looks more like this first and second layer, whereas heterochromatin is more densely staining, and therefore it is more tightly packed, and it contains DNA with these higher levels of chromatin organization. These two terms, euchromatin and heterochromatin, are very important terms, and just make sure that you note which one is which. Euchromatin is the light staining, the less dense DNA, whereas heterochromatin is the more darkly staining, more densely packed DNA. So what scientists now understand is that the ability to transcribe a particular gene on the DNA is very, very strongly influenced by how tightly packed the DNA is in that particular region of the chromosome. So if you have a situation like this, factors such as RNA polymerase and transcriptional regulators are much more likely to be able to bind than if the DNA looks like this or is even more tightly packed. So I'm going to introduce three different processes that regulate this chromatin structure and therefore can affect whether a particular gene is transcribed or not. These three are chromatin remodeling, modification of histone proteins, and DNA methylation. So first, I'll talk about chromatin remodeling, which is the repositioning of nucleosomes on the DNA. This is an essential process in order to expose different regions of the DNA for binding by factors such as RNA polymerase and transcriptional regulators. So what will often happen as you try to activate transcription in a particular region of a eukaryotic chromosome is that one factor will bind, say this might be a binding site for a transcription factor, and it will bring in other factors which help to free up that area of the chromosome. And so what has happened here is that a transcription factor will bind, and it will bring in a second factor called a chromatin remodeling complex. This chromatin remodeling complex will actually move nucleosomes around. So it's something that actually moves nucleosomes out of the way so other factors can bind to that region, such as RNA polymerase and all of the associated basal transcriptional apparatus that is needed to transcribe a gene in eukaryotes. Another thing that regulates the chromatin structure are modifications that are put on the tails of the histone proteins. So here is depicted a nucleosome. So this has 8 different histones all bound together with DNA wrapping around them. The histones all have two main parts to them. They have this sort of globular region, which is the region that makes up the main part of the nucleosome that interacts with the other histone proteins. And then they also have these tails, which are at the N-terminus of the protein, and those tails kind of hang out of that main globular part of the protein around which the DNA wraps. So it turns out that the tails of the nucleosomes can receive modifications. So these are proteins, and only particular amino acids are able to get modified. So these N-terminal tails of these proteins have a lot of lysine and arginine amino acids, and these are some of the main ones that get modifications put onto them. There are different types of modifications that are put on, and the different types of modifications lead to different effects in how tightly the DNA wraps around those nucleosomes. Some of the modifications are shown here. So some of these amino acids can be methylated, others can be acetylated. Those are the two main modifications, but there are several other things that can also happen, including phosphorylation and ubiquitination of these nucleosomes. Overall, the modifications that are put on the tails of these nucleosomes create something that is called the histone code. And it is a code that is read by the cell, and the overall code determines how tightly the DNA wraps around those nucleosomes. I'm going to talk about two of the types of modifications that are used in the histone code. The first modification is acetylation of histone tails. This one is probably the best understood modification of histone tails, and it involves lysine amino acids. And so here is the structure of the lysine amino acid. It's the NCC is the backbone of the protein, and the side chain is shown here. This is a lysine side chain. So an acetyl group is the addition of these two carbons and oxygen. This is added to that nitrogen at the end, and it actually changes the charge of that side chain. So if you do not have an acetyl group on the end, the lysine residue is positively charged, and once you add the acetyl group, then it is no longer positively charged. People have theorized that possibly the charge has something to do with how this functions. And so if you see the DNA here is shown in gray and the histones are shown in pink, and DNA is negatively charged, and so you could imagine that those histone tails would tend to bind pretty tightly to the DNA when the lysine side chains do not have the acetyl group, when they're positively charged, that electrostatic interaction. And when the lysine gets the acetyl group put on, that positive charge is then removed from that side chain, and that electrostatic interaction between the DNA and the histone tails would no longer be present. So the result of acetylation is that the DNA is less tightly bound in with the nucleosomes, and that's portrayed here by having these loose ends of the protein arms. The acetylation of lysine side chains is reversible and is also regulated by enzymes. And so the enzyme that adds an acetyl group onto the lysine side chain of a histone is called a histone acetyltransferase, and that is designated by this abbreviation HAT for histone acetyltransferase. And then the enzyme that removes the acetyl group and makes that lysine go back to its normal state is called an HDAC for histone deacetylase. So this depicts the overall effect that acetylation has on the chromatin. So if you have unacetylated histone tails, that DNA is going to be wrapped very tightly within these nucleosomes, and this would make a rather heterochromatic chromatin environment. Those histone tails can have acetyl groups added to their tails, and that positive reaction is done by histone acetyltransferases. And the result of adding those acetyl groups is that the entire DNA is now more relaxed. There is more space between the nucleosomes, and it is just less dense overall. Acetyl groups can then be removed again by these histone deacetylases or HDACs. Because histone acetyltransferases lead to this open chromatin conformation that allows transcription, we would actually say that HATs are transcriptional activator proteins. And since HDACs lead to this tighter chromatin conformation, we would say that an HDAC is a transcriptional repressor. The second type of modification I'd like to talk about with regard to the histone code is the methylation of histones. So lysines are also the amino acids that are modified by methylation, and this is shown here. So here is the lysine. It's a slightly different type of model, but there's the N, the CC, and the COOH over here. And this is the side chain for the lysine, ending with a positively charged nitrogen. And so you can add one methyl group to that nitrogen, or you can add two methyl groups, or even three methyl groups. This would be mono, di, or tri-methylated lysine. The enzyme that adds methyl groups to lysine side chains is called histone methyltransferase. And there's another enzyme that removes the methyl groups that is called histone demethylases. So the effect of methylation of lysines on histones is kind of mixed. It actually depends on which particular lysines in that tail are methylated. And so if we look, for example, at histone H3, the N-terminus of that protein is shown here. So A is the first amino acid, then T, then R, then K. And each amino acid is numbered according to its position from the N-terminus. And so lysine K4, for example, if that particular one is methylated, you tend to loosen the region of chromatin. It becomes more euchromatic. That increases the accessibility of the DNA and leads to gene activation. In contrast, lysine 9 and lysine 27, if those ones are methylated, those particular ones lead to gene silencing and a tightening up of the DNA with the histones, making it more heterochromatic. The last type of modification I'd like to talk about is the methylation of DNA itself. So DNA can be methylated on a particular spot. It can be methylated only on cytosine bases. So here is the normal cytosine, and if that DNA is methylated, it gets put right here on that particular base. In addition, only particular cytosines are able to be methylated, and those are ones that are followed by a G. So you have to have a C followed by a G, and if you have those two, then possibly that C could actually be methylated. C's followed by G's have a particular name. They're called CPGs, and sometimes you'll see a whole region that has many C's followed by G's being called a CPG island. So DNA methylation is a pretty strong effect to cause the DNA to be transcriptionally repressed. It makes the region of the chromosome more heterochromatic, more tightly wrapped in their nucleosomes, and this type of modification is mostly associated with very stably inactivated regions of the DNA. So these would be regions of the DNA that the cell usually does not anticipate using again in its lifetime. An example of how this might be used is during development, as certain cells start to differentiate into different types of cells. One group of cells may become, for example, a muscle cell, and another group of cells may become a neuron. And that muscle cell is going to permanently inactivate the regions of its genome that have to do with making a neuron, because it plans to never do that again. And so it might methylate regions of the genome that have to do with functioning as a neuron, for example. DNA methyltransferases are the enzymes that add those methyl groups to those cytosine bases. So these two modifications that we talked about the histone code, which can involve methylation of the tails or acetylation of the tails of the histone proteins, and methylation of the DNA itself, are what we call epigenetic marks. And epigenetic marks just mean that those are changes that are made to the DNA that can affect how that region of the genome is used, but those changes do not involve a change in the actual DNA sequence itself. So I like to think of this as sort of, you have the underlying order of the bases, and that is really not changing here. But what you can change is you can change the icing on top of that, of those DNA bases, the icing on the cake that determines how that region of the chromosome is being used. One final point I'd like to make is that the histone code and DNA methylation are very tightly linked to each other. And so two examples are shown here, and please realize I don't want you to learn all of the details of all of these factors. I just want you to understand this basic thing. So for example, if you get methylated CPGs near a gene, that's going to recruit a particular protein called methyl CPG binding protein 2, and this then recruits a complex of proteins that includes HDACs or histone deacetylases. And so the methylation of the DNA also led to a change in the histone code. Likewise, here's an example of a place where the histone itself gets methylated. If lysine 9 on histone 3 is methylated, this is repressive for transcription. And what happens here is that methylation recruits a protein called HP1, which then brings in DNA methyltransferase. And so DNA methyltransferase is the enzyme that will actually methylate the DNA itself. And so here we had methylation on a histone leading to DNA methylation. To wrap up, I'd just like to summarize the differences then that we have talked about between euchromatin and heterochromatin. So euchromatin is much more loose, it is less densely packed, and it tends to be that active genes are present in the euchromatic regions. Histone acetyltransferases are often recruited to euchromatin, the histone tails become acetylated, and certain lysines on the tails of the histones are methylated as well. Transcription factors can get bound to this region of the chromosome since it has more space between the nucleosomes, and often those transcription factors would recruit factors like SWI/SNF, which is a chromatin remodeler that can then move nucleosomes around to expose binding sites for things like RNA polymerase and the basal transcriptional apparatus. In contrast, heterochromatin is very highly condensed. Definitely regions of the genome that have a lot of repeats tend to be heterochromatic, and it tends to be regions also of the genome that don't have a lot of genes in them. In these regions, histone deacetylases tend to be recruited, the histone tails are not as acetylated, and certain types of histone methylations are present. The DNA is methylated in heterochromatin, and DNA methyltransferase is what methylated those CPGs. There is a region in between the two of these, and so you can imagine that not every gene is either on all the time or off all the time. There are some genes that we might turn on and off depending on a signal from the environment. So in the middle here is something we would call facultative heterochromatin, and that's simply a state that could easily be induced to become euchromatic and become transcriptionally active.